Welcome to Project SCORE, which is a publicly available online database from the Cancer Dependency Map at the Wellcome Sanger Institute and can be used to identify cancer cell dependencies and prioritise therapeutic targets using data generated from CRISPR-Cas9 whole genome dropout screens. Currently, the database allows the user to explore over 7,000 fitness genes from over 300 cancer cell lines across 19 different tissue types. So if you have a gene, cell line model, or a cancer type of interest, you can search for that interest from the search bar here on the homepage. Or if you want to explore the data in a more unbiased fashion, you can explore all fitness scores or all target priority scores here from the homepage. So for example, let's select Explore Target Priority Scores. And these are scores that have been assigned to all fitness genes to nominate promising therapeutic targets. So we can filter the scores based on a cancer type. We can also look at a pan-cancer analysis. So for example here, let's select breast carcinoma. We can then further filter the target priority score based on the strength of the associated biomarker and cell line fitness effects. So for example, we can decrease or increase the minimal fitness score, and we can also adjust the false discovery rate. And we can make any updates by selecting Update. And here we can see that all genes or targets have been grouped into tractability buckets. And the most promising tractable targets are grouped into Group 1 buckets. As this is breast cancer, we can see ERBB2, ERBB3, and PIK3CA, which are all promising therapeutic targets in breast cancer. If you have a specific gene of interest, we can return to the homepage and you can search for that gene in the search bar. So for example, let's look for Warner, which is a DNA helicase associated with DNA damage and transcription. So here we can see on the gene view page, we have the fitness summary, and we know that Warner is associated with loss of fitness in 22 cell lines across 6 different cancer types. For more information on the gene of interest, we have external links provided in the top right. We've also recently introduced a target priority score panel, and we know that Warner is a promising therapeutic target in colorectal, gastric, and ovarian cancer. We can see the total priority score as well as the rank of that priority score in each cancer type. We can also examine the strength of the genomic biomarker and the associated fitness effects. We've also recently introduced a target profile panel where we can see associations with known drugs and cancer hallmarks. So for Warner, we have no current drugs available and there is no association with cancer hallmarks. We can also see information on tractability, and we know that Warner has both predicted precedence and discovery precedence. This information is directly extracted from the Open Targets portal, which can be accessed via a direct link here on the top right. We also have the fitness plot and fitness table if we scroll down, and we can also filter these tables and plot by cancer type. So for example, let's select colorectal carcinoma. And we can see here that Warner has a significant loss of fitness effect in multiple different colorectal cancer cell lines. We can select cell lines of interest directly from the fitness table here. So for example, let's select RKO. And these cell line models can also be searched for from the homepage. So we can see more information on the cell line model. And we can also see direct links to multiple genomic datasets from cell model passports and GDSE. So if we scroll down, we can see our fitness plot and fitness table, and we can see all genes that are associated with a significant loss of fitness in the RKO cell line. This data is also represented in the fitness table. So for more information on how we generated our data, you can access this information from the documentation tab, where you can find all the information that you need on how the data was analysed and interpreted. We also have a Downloads tab where you can freely access data generated in the Wellcome Sanger Institute, the Broad Institute, as well as our integrated datasets. So for more information or for feedback, please contact us at depmap@sanger.ac.uk, and thank you for listening.